All right, let's get right into it. If you're gearing up for Professor Pasquinelli's BIM 100 final, you are in exactly the right place. We're going to do a quick and dirty review of the absolute must-know concepts from those first 4 lectures. Let's do this. Look, instead of just trying to cram a bunch of facts into your brain, the best way to make this stuff stick is to think of it like a story. It's the story of a cell and how it solves its biggest, most fundamental problems, so it can, live and make more of itself. Framing it this way will seriously help you connect all the dots for the exam. So, what are these big challenges? We're going to cover 3 of them from your first few lectures. First, you've got the blueprint problem. How on earth do you store the instructions for life? Then, there's the packaging problem. How do you fit a massive blueprint into a space that's microscopically tiny? And finally, the copying problem. How do you make a perfect, flawless duplicate of the whole thing? Okay, let's tackle that first problem. Before a cell can even think about doing anything else, it needs a super stable, super reliable way to store its master plan. And nature's elegant solution to that? It's DNA, of course. But what is DNA really? Well, it's made of these basic building blocks called nucleotides, and you absolutely have to know the 3 parts for your final. You've got a phosphate group, that's the energy in the backbone. You've got a 5-carbon sugar, which in DNA is deoxyribose, and then you have the nitrogenous base. That's the actual letter in the genetic code, the A, T, C, or G. Okay, this right here, this is gold for the exam. It's Professor Pasquinelli's super practical 3-step method for naming any nucleotide. Seriously, just follow the steps. 1. What's the sugar? Deoxyribose for DNA. 2. What's the base? A, G, C, or T. And 3. How many phosphates? So something like deoxyadenosine triphosphate. If you can follow these 3 steps, you are going to nail these questions. Now, those bases come in 2 different flavors. You got your purines and your pyrimidines. A super simple way to remember this is that purines, that's adenine and guanine, they're bigger. They have 2 rings in their structure. Pyrimidines, cytosine and thymine, are smaller with just 1 ring. And this difference in size is actually super important for making sure the DNA double helix has that perfect uniform shape. Okay, now this is a concept that seems small, but trust me, it's huge. A DNA strand has a direction. It has a 5' end and a 3' end. But here's the kicker. The 2 strands of the double helix run in opposite directions. They're anti-parallel. This might sound like a tiny detail now, but it has massive, massive consequences when we get to our 3rd challenge, the copying problem. Just file that away. All right, moving on to challenge number 2. So you've got your blueprint, and in humans, that blueprint is about 2 meters long if you stretched it all out. Now you have to somehow cram all of that into a nucleus that's about 6 micrometers wide. That, my friends, is a serious packaging problem. So how does the cell pull this off? With a substance called chromatin. The best way to think about it is this. Your DNA is like a super long piece of thread, and these proteins, called histones, are like tiny little spools. By wrapping the DNA thread around these histone spools over and over again, the cell can condense its genome with incredible efficiency. Now pay attention here, because this is a super high-yield concept for the final. Chromatin isn't all the same. It comes in 2 main states. First, you have euchromatin. Think U for true or good. It's open, it's loose, and the genes in that area can be read. They're active. This is linked to histone acetylation. Then you have heterochromatin. This stuff is shut down. It's tight, it's condensed, and the genes there are silent. This is linked to histone methylation. Active versus silent. That's the big idea. So you might be wondering, how do we even know this stuff is real? Well, scientists figured it out with clever experiments, like the DNA-Cy protection assay. The logic is actually really simple. If you dump in an enzyme that chews up DNA, like DNA-Cy, it can only get to the DNA that's open and exposed, the euchromatin. The tightly packed heterochromatin basically acts like armor, protecting the DNA inside. So if the DNA survives, it must have been hiding out in that silent, protected state. Okay, so the blueprint is stored and it's all packaged up neatly. Now for the final and maybe the most complex challenge from these early lectures. How does the cell make a perfect copy of its entire genome every single time it needs to divide? This is all about DNA replication.
The most fundamental principle you gotta know about replication is that it's semi-conservative. That sounds fancy, but all it means is that when the cell unwinds the double helix, each of the two original strands is used as a template to build a brand new one. So you end up with two new DNA molecules that are identical, and each one is made of one old strand and one new strand. It's just a beautiful, simple system for making a perfect copy. Now, this whole process doesn't just happen by itself. It takes a whole team of specialized enzymes, a real molecular all-star lineup. You've got helicase, that's the unzipper, topoisomerase runs ahead to relieve the stress of unwinding, primase comes in and lays down a little RNA primer like a starting block. Then you have the star player, DNA polymerase, which actually builds the new strand. RNase H cleans up by removing the primers, and finally DNA ligase is the glue that seals up all the gaps. Okay, remember way back when we talked about the two DNA strands being anti-parallel? Well, this is where that comes back to bite us. Because DNA polymerase can only build in one direction, one of the strands, the leading strand, can be copied in one beautiful, smooth, continuous piece. But the other strand, the lagging strand, is pointing the wrong way, so it has to be copied backwards in a bunch of little stitched-together pieces called Okazaki fragments. And this is exactly the kind of format you can expect on the final. You'll get a question like, what would happen if you added a drug that blocks helicase? Or what if DNA ligase was mutated? To get those questions right, you have to know what each member of that enzyme team does. Knowing their specific jobs is absolutely crucial. But wait a second, that lagging strand creates one last sneaky little problem. Because of the way it's made in pieces, and because you need a primer to get started, there's no way to copy the very, very end of the chromosome. This means with every single round of replication, the chromosome would get a little bit shorter, and a little bit shorter, which would eventually be a total disaster. But the cell has an ingenious fix. And that fix is an absolutely amazing enzyme called telomerase. Its whole job is to protect and extend the very ends of the chromosomes, which are called telomeres. And what's so wild about telomerase is that it's a reverse transcriptase. It actually carries its own little RNA template inside of it, and it uses that template to build DNA, adding these repetitive little cap sequences to the chromosome ends. It works in a really slick three-step process. First, it just binds to the tip of the chromosome that's hanging over. Second, it uses that internal template to extend the DNA, adding its little repeat sequence over and over. And third, once that's done, the normal replication machinery can come in and fill in the other side. And just like that, the chromosome end is longer and protected. Problem solved. So, when you zoom out, the big takeaway from these first 4 lectures is that biology isn't just a random list of facts to memorize. It's a story of problems and really elegant solutions. The cell uses the beautiful structure of the double helix for storage, the clever system of chromatin for packaging, and a whole precision team of enzymes for copying. And to leave you with one last thing to really chew on for the exam, something that connects all these ideas. In most of our adult cells, telomerase is turned off. That's part of what limits their lifespan. But almost all cancer cells figure out a way to turn telomerase back on. So the question is, why is flipping that switch back on so absolutely critical for a tumor cell to become immortal? Think on that. Good luck with your studying. You're going to crush this.